Good morning. Today we'll be looking at bacterial cell count and viability measurement using hemocytometer. This is a side view of a hemocytometer, and we have already placed a cover slip on top of one of the chambers. There are two chambers in a hemocytometer and we only use one of them to load the sample. And the reason for placing a cover slip on top of the chamber is to create a defined volume. Normally, when you perform an experiment with hemocytometer, it is to determine the number of cells per ml of sample. In order to do that, you need to create a defined volume. By placing a cover slip on top of one of the chambers, you create a depth of 0.1 millimeter over an area of 1 millimeter squared. This can be used for counting the number of bacteria or cells per ml of sample. Now let's take a look at the hemocytometer grid. It's divided into 9 squares, 5 central squares and 4 corner squares. The 5 central squares will be used for counting the RBCs, and the 4 corner squares will be used for counting the WBCs. We for direct microscopic count of bacteria or yeast or other cells like sperm, we commonly use the 4 corner squares, also called as the WBC squares. Now let's take a look at the bacterial cell number calculation. Each of the two grids of the hemocytometer, the chamber 1 and chamber 2, is a square with a size of 3 millimeter. The square is divided by 3 vertical and horizontal equidistant lines that form 9 squares with a size of 1 millimeter. The area of each square is therefore 1 millimeter squared. Placing a cover slip on the hemocytometer forms a chamber that is 0.1 millimeter high. So the cell suspension volume will be 1 millimeter squared into 0.1 millimeter, which is equal to 0.1 millimeter cube. To know the number of cells in 1 ml, it will be enough to divide the number of cells counted in a square by 10 to the power minus 4 milliliters, which is by multiplying the number of cells by 10 to the power 4. Now let's take a look at the bacterial cell count formula. This is not just for bacteria, it can be used for yeast or other cells. So the formula for calculating cell number using hemocytometer is N into 10 to the power 4 into F cells per ml. The cells per ml is actually the unit. Always remember to use them. N is the average number of cells per one of the corner squares. So you can count from all the 4 corner squares and take an average, and that will be your N value. N is the average number of cells counted in the different squares. We only use the 4 corner squares to do the count. If the cells are diluted because they are too dense or with trypan blue for viability test, then the number must be multiplied by the dilution factor. So F is the dilution factor. For example, if the cells are diluted 1 is to 2, but putting an equal volume of cells and trypan blue, then the formula will be N into 10 to the power 4 into 2. Normally that's the dilution that we use. Viable cells calculation. On top of determining the number of cells per ml of sample, it is also important to calculate the percentage of viability. The formula for calculating the percentage of viable cells is by dividing the number of viable cells by the number of total cells and multiplying by 100. So viability is always given in percentage. So percentage viable cells is equal to number of colorless cells divided by number of total cells into 100. So before we load the sample, we take 100 ml of our sample and mix it with 100 ml or equal volume of trypan blue. So there is something special about trypan blue. It only stains the dead cells. So they are negatively charged in the bacteria, or cells are also negatively charged, so they don't readily take the stain. I mean, living cells do not readily take the stain, but dead cells or damaged cells usually will get stained. So when you're making the count, you have to count the colored cells, which are like the damaged or dead cells, and the colorless cells separately. So when you do that, you could apply that to this formula. So percentage viable cells is equal to number of colorless cells. So the colorless cells are actually the viable cells, divided by total number of cells. Herein you will add both the colored cells as well as the colorless cells, and this should be multiplied by 100 because the viability is always given as a percentage. Let's take a look at the procedure. First, place 100 ml of cell suspension in an Eppendorf tube, to which we add equal volume of 0.4% trypan blue to the cell suspension to obtain a 1:2 dilution, and mix by pipetting up and down. Then we incubate the mixture for less than 3 minutes at room temperature. If cells are counted after approximately 5 minutes, viability will be inaccurate due to cell death. With a cover slip already in place, fill one side of the hemocytometer chamber with the cell suspension by placing the tip of the pipette at the notch. Typically, each side will take 10 to 20 microliters. Place the hemocytometer on the stage of a light microscope and focus on the cells. Each side of the hemocytometer contains multiple squares. Count all squares clear and blue in each large square in each corner of the hemocytometer. Each large square contains 16 small squares. In each large square, count cells that are on the border lines on two sides only. Keep track of the number of blue cells separately as well as part of the complete number of cells. Blue cells are the non-viable cells. Calculate the number of bacterial cells per ml and percentage of viable cells using the formula and tabulate the results. And thus, by performing this experiment, you'll be able to calculate the number of bacterial cells per ml of sample and the percentage of viable cells. Thank you.